Hi everyone, this is going to be the second video in the bioenergetics topic for advanced level biology. In the last video we looked at an introduction to this topic and we classified organisms as being autotrophs or heterotrophs. In this video we're going to be looking at a specific form of autotrophy, photosynthesis, and we're going to be focusing specifically on the biochemistry of photosynthesis. So the light-dependent reactions and the light-independent reactions are the two categories of biochemical reactions involved in photosynthesis. So as we go through this lesson, we're going to be drawing a distinction between the light-dependent and the light-independent reactions. Now, these are essentially arbitrary distinctions, but it makes it easier for us to study and to talk about the biochemistry of photosynthesis if we divide them. If we divide all of the different biochemical processes involved in photosynthesis into those two broad strands, if you like, or two broad categories of light-dependent versus light-independent. So the light-dependent reactions of photosynthesis take place in the chloroplast. Specifically, they take place in the thylakoid membranes of the grana of the chloroplast. Now, again, to remind you, the grana are these stacks of flattened membrane-bound vesicles called thylakoids. So these are membrane-bound vesicles and they are stacked in these arrangements called grana. Now the word grana is the plural, granum is the singular. So here we see the thylakoid membranes of this granum. Between the grana you have these thin membranes or thin membrane connections between the thylakoids of separate grana called lamellae. So the light-dependent reactions begin when energy in the form of visible light from sunlight is captured by the light-absorbing pigments located within the grana. So light energy travels as electromagnetic waves, but electromagnetic waves can be modelled as though they are particles carrying a specific discrete quantity of energy, and these particles are referred to as photons. Now, I'm not going to get into wave-particle duality here because that's a subject for A-level physics, but essentially it's enough to state here that light and electromagnetic waves in general can be modelled in two different ways. They can be understood and investigated in two different ways. The first of these ways is perhaps the way in which you have been talking about them and describing them at GCSE. And so you often speak about the electromagnetic spectrum in terms of a range of different wavelengths or frequencies. And what you're doing there is you're modelling electromagnetic waves, as oscillations of electric and magnetic fields which propagate through space. Now that's one way of modelling or understanding waves, electromagnetic waves like light. A different model, which is just as valid, is to understand it in terms of discrete packets of energy, like particles, and these packets of energy are called photons. And so, for the purposes of this discussion of photosynthesis, it is more useful to model the energy carried by light as being composed of discrete packets of energy called photons. So the energy content of a photon of light depends upon the wavelength or the frequency of the light. And so the shorter the wavelength, typically, the higher the photon energy. Now the energy associated with a photon of light is called a quantum of energy. This comes from the Latin quantum, which means how much. And again, this goes back to some of the foundational ideas of the science of quantum theory, where energy carried by phenomena like light, for example, and also by electrons, is quantized. It is discrete in the sense that it can take one value or a higher or lower value, but nothing in between. Now again, we're not going to get into the specifics of quantum theory here, but it's enough to say that the energy associated with one of these packets of light is referred to as a quantum of energy. And so in this respect, blue light has a relatively short wavelength and therefore a relatively high amount of energy carried by each photon, or a relatively high quantum of energy carried by each photon, and correspondingly red light has a relatively long wavelength or a low frequency, and therefore each photon of red light carries comparatively less energy. So plants are able to absorb a wide range of wavelengths because they contain a variety of pigments with different structures and absorption properties. Now this again goes into areas of quantum theory and also chemistry which fall outside of the bounds of advanced level biology. Essentially, the color of a substance, particularly a colored substance or a pigment, is dependent upon the arrangement of atoms in the molecules of that pigment. The bonds between those atoms and the electrons in those bonds can absorb and emit light of different frequencies, and the specific arrangement of atoms and bonds is what gives that material its ability to absorb and reflect and transmit specific frequencies of light, and in turn that's what gives that material its specific colors. Now, as we'll see, the pigments, the colored compounds found in plants which are responsible for absorbing various different frequencies of light are quite varied. At GCSE level, you perhaps thought of the pigments associated with photosynthesis as simply being chlorophyll, the green pigment. And again, it's green because it reflects the green wavelengths while absorbing the blue and the red. But as we'll see, there are a range of different photosynthetic pigments. So again, visible light represents only a small fraction of the full spectrum of electromagnetic radiation produced by, for example, the sun. So this will be familiar to you from GCSE physics perhaps. So visible light, if we represented the electromagnetic spectrum as various ranges of wavelengths corresponding to the different types of electromagnetic waves, visible light would be a very short range, somewhere in the middle between the ultraviolet at the higher end of frequencies or lower wavelengths and the infrared or heat radiation at the longer wavelengths and the lower frequencies. So visible light comprises a range of wavelengths which is actually quite a narrow range when compared to the overall electromagnetic spectrum. So the plant pigments that absorb light energy with wavelengths that fall within this visible spectrum are the ones which are responsible for transducing that energy for photosynthesis. So the pigments can be extracted, and perhaps you've done a practical of this sort yourselves. It can be extracted from finely chopped leaves by grinding the tissue in a solvent like acetone. So the way that you would then go on to separate the mixture of pigments is to do so by some form of chromatography. So for example, we can use paper chromatography for this. And by doing this, you will find that the pigments found in a green looking leaf are actually of a variety of different colors, and these correspond to various kinds of photosynthetic pigments. Now some of these you'll be familiar, particularly with chlorophyll from GCSE biology, but the other ones perhaps you may not be so familiar with. So the principal plant pigments found in green plants are the chlorophylls. There are two variants shown here, chlorophyll A and chlorophyll B, but also the carotenoids, carotene and xanthophyll, which are the ones being shown here, which have the more orangey brown colours, as you can see. Now again, the role of all of these pigments ultimately is to capture the sunlight energy, and each of these pigments reflects a different range of wavelengths, which are the ones, the colours that the pigments appear to us to be, and the other wavelengths are absorbed by these pigments. So the photosynthetic pigments embedded in the thylakoid membrane, there aren't just different varieties of them, but they are arranged in a certain manner. Now, they tend to be arranged, the molecules of these photosynthetic pigments tend to be arranged in clusters, organized clusters, which are referred to as photosystems. So this diagram here represents a kind of, it's not the way that the photosystems themselves are necessarily organized in reality, but it's a good representation that allows us to understand how they work. So each photosystem consists of a number of specialized chlorophyll molecules which are arranged in a particular way, together with an even more specialized chlorophyll molecule called a reaction center, and they're arranged in a complex like this, which is often referred to as an antenna complex. So this whole thing here that's being shown is a photosystem. Now the antenna complex is composed of various kinds of photosynthetic pigments, carotenoids, chlorophylls, and so on, and these are designed in this arrangement so that they can capture and pass the various wavelengths of electromagnetic energy to a specialized chlorophyll molecule called a reaction center. So the antenna pigments, essentially their function is to gather sunlight of various different wavelengths and then funnel that light, hence the shape of this funnel arrangement here. Again, this is a kind of metaphorical way of showing the antenna complex and the reaction center. The job of the molecules of the pigments in the antenna complex is essentially to funnel various wavelengths of electromagnetic energy to this chlorophyll molecule called the reaction center. So the light energy comes in, it's absorbed and then passed between these specialized chlorophyll molecules, ultimately finding its way to the reaction center. So in this way, the chlorophyll molecule at the reaction center ultimately receives all of the absorbed energy from all of these different chlorophyll molecules in the antenna complex. And this whole photosystem that we're seeing here is embedded in the thylakoid membranes of the grana in the chloroplasts. So this is what is ultimately responsible for capturing the sunlight energy. Now, it's not enough to just talk about how the sunlight energy is captured. We then need to understand what is done with that energy which is funneled by the antenna complex pigment molecules to the reaction center. How does that energy ultimately find its way into a usable form of energy in the form of glucose in the plant? So for that, we need to start by looking at the details of the light-dependent reactions. So the thylakoid membranes of the grana contain two types of photosystem. We call these photosystem 1 and photosystem 2, or PS1 and PS2 respectively. Now, we can show what happens with the light energy which gets captured by these two different kinds of photosystems on a diagram which maps the relative energy levels of these photosystems and the molecules which are associated with them in the thylakoid membrane. Now, what we have here is the two photosystems. So we have they're kind of oriented upside down with respect to how they were in the previous slide. This is the antenna complex for photosystem 2. This is its reaction center. Again, this is the antenna complex for photosystem 1. This is its reaction center. So light in this diagram is coming from underneath in this direction here to the antenna complexes. The antenna complexes then funnel that light energy to the reaction centers. Now, as we can see here, on the left-hand side, we have an axis here which says redox potential. Now, for those of you who don't do advanced level chemistry, you can think of redox potential as essentially the level of potential energy at which you find the photosystems and the various other molecules which are involved in the light-dependent reactions which are embedded within the thylakoid membranes. So, the two photosystems, they do differ from each other in a number of ways, but primarily by virtue of the wavelengths of the light that the reaction center chlorophyll molecules absorb, and also the photosystems are, as shown here, are at different redox potentials or different energy levels. Now, the redox potential refers to the energy content of the electrons in these photosystems, particularly the electrons in these reaction centers. Ultimately, it's the electrons which absorb that sunlight energy, those packets of energy called quanta of energy carried by the photons of sunlight. So we're going here from a lower electron energy level down here to a higher electron energy level up here. So we're going to make use of a diagram like this to show what happens to the sunlight energy, how it's passed on between molecules in that thylakoid membrane, and what is done with that energy. So that's kind of just introducing the way that we're going to represent that in a diagrammatic way here. So I mentioned that the photosystems 1 and 2 are not the only molecules that you find in the thylakoid membranes. You also find a number of other molecules which are involved in the light-dependent reactions. So you have primary electron acceptor molecules, which are being shown here. Now notice the primary electron acceptor molecules are found, again, these are all embedded within the thylakoid membranes, but they are found at different redox potentials or at different electron energy levels. You also find carrier molecules, which form an electron transport chain. Now, the term electron transport chain by the end of this topic will become very familiar to you, because you'll find that there are electron transport chains involved not just in photosynthesis, but also in respiration as well. Now, as we'll see later in this topic and throughout this topic, again, it's the electrons ultimately which carry that energy from one molecule to another. So we have not just the primary acceptor molecules, we also have carrier molecules which form electron transport chains. So all of these molecules, the photosystems, the reaction centers of those photosystems, the primary acceptors, and the molecules which make up the carrier molecules which make up the electron transport chains, these are shown at their respective electron energy levels on this diagram. So the way that this works is you have not just the primary acceptor molecules. As we'll see, electrons are transported between the photosystems to the primary acceptors via the electron transport chain carrier molecules, ultimately to a molecule called ferredoxin, which is an electron acceptor. So all of these components are the ones required for the light-dependent stage of photosynthesis. And again, they are ordered on this chart with respect to their respective energy levels or redox potentials. So how does this work then? Well, let's start with the photosystems again. So the start of the light-dependent reactions is obviously when light comes in and strikes the grana, or the thylakoid membranes of the chloroplasts. The antenna pigments, the antenna complexes containing those pigments, the chlorophylls, the carotenoids, and so on, will then absorb light energy, and the electrons in those pigments are promoted from a lower energy level to a higher energy level. Again, this is goes back to the principle of the conservation of energy. If you think back to your GCSE chemistry, when you drew those electron shells or energy level diagrams for atoms, it was the innermost shell which contained the lowest energy level electrons, and then you got to higher and higher energy levels as you go outward from the nucleus. Well, the same thing applies to molecules. Molecules are atoms which have been connected to each other by chemical bonds, so it's an arrangement of atoms with chemical bonds between them. But in all cases, those chemical bonds involve electrons. So in atoms, electrons are arranged in energy levels around the nucleus of each atom, but in molecules, electrons can also be arranged in bonding orbitals, which themselves have electron energy levels. So when electrons in an atom or a molecule absorb energy in the form of a packet or a photon of energy, a photon of light, for example, that energy has to go somewhere, because it's the principle of conservation of energy. Energy cannot be created or destroyed. It can only be transferred from one object or one system to another. So when an electron absorbs a certain amount of energy, it goes from that lower energy level to a correspondingly higher energy level, and the difference between those energy levels is exactly the same as the amount of energy that that electron has absorbed. So when the electrons in the pigment molecules in the antenna complexes of these photosystems absorb those photons, those packets of energy, they are excited. That's the kind of technical term for them. They are excited from a lower energy level to a higher energy level. And so this is what we refer to as photoexcitation. Now, the way that it works is that two photons are absorbed. So energy is funneled to each of the reaction centers when the antenna complexes absorb sufficient quantities of energy. And this happens by means of the absorption of two photons of energy. Those photons of energy are then transferred via the antenna complex to the photosystems, to each of the reaction centers in those photosystems, and then this causes electrons to be released from those reaction centers, because the electrons are now at a higher energy level than they were in those reaction centers. So a single electron is released from each reaction center for each photon which is absorbed, and so we have two electrons which are promoted from this lower energy level and this lower energy level to a higher energy level, because they have absorbed a certain amount of energy. So the released electrons are then accepted by those primary acceptor molecules. And so again, we've got two electrons sitting at this energy level here in photosystem 2. They absorb two packets or quanta of energy from the two photons which are funneled to that reaction center via the antenna complex pigment molecules. Those electrons then absorb that energy, so they go from this energy level or this redox potential to this energy level. At this energy level in the thylakoid membrane, they are accepted by the primary electron acceptor molecules. The same thing happens for photosystem 1. So the release of the electrons from the photosystems creates what's sometimes referred to as electron holes at the reaction centers. Now these need to be filled if photoionization is going to occur again. Now again, the word ionization refers to whenever a molecule or an atom which is uncharged, electrically neutral overall, has lost or gained electrons and therefore formed a charged, an overall charge, or formed an ion. In this case, it is photoionization because the reaction center has lost electrons and therefore this has left two positively charged holes, if you like, where those electrons used to be. Now, as we'll see later on, those holes are filled by electrons, and they have to be before the photosystems can be excited again by the absorption of two more photons. So the electron hole at photosystem 1 is filled because electrons pass along a chain of carriers from the primary acceptor for photosystem 2. So this is what happens here. Both of these, and again, this is a continuous process whenever light is available. I'm kind of breaking it down in this order, in this fashion, but potentially you could start from any point in this process because it is a continuous process. So once the electron hole in photosystem 1 has been formed by the photoexcitation, the photoionization of the reaction center of photosystem 1 due to the absorption of these two photons, those two electrons are excited and move to this primary acceptor molecule in the thylakoid membrane. This leaves an electron hole, two electron holes, in photosystem 1, but these are filled because the electrons which were excited and photoionized from photosystem 2's reaction center are passed from this primary acceptor molecule down this chain, this electron transport chain of carrier molecules, and at the end of that, the final carrier molecule in the electron transport chain transfers that pair of electrons to fill the electron hole in photosystem 1. So this happens continuously. Now the flow of electrons along this electron transport chain, as you can see, each one of these carrier molecules is at a successively lower electron energy level or a successively lower redox potential. Now the differences between these the differences in redox potential between these successive carrier molecules, that difference in energy has to go somewhere. It doesn't just disappear. Again, think of the law of conservation of energy. And so in fact, that energy difference from here to here, for example, or from here to here, the electron is losing energy, but the energy is going into the production of ATP from ADP. And again, in the biological molecules topic, we did a whole lesson on the structure of adenosine triphosphate and the fact that it's referred to as the energy currency of cells. Now again, later on in this topic we're going to see how ATP is produced during respiration, but for now we can see that ATP is actually formed directly during the light-dependent reactions of photosynthesis. So the flow of electrons along this electron transport chain from the primary acceptor for photosystem 2 to the reaction center for photosystem 1 is actually used as a source of energy for the production of ATP from ADP and inorganic phosphate. So the formation of ATP as the photoexcited electrons pass along this chain, this is sometimes referred to as photophosphorylation. It is phosphorylation of ADP because you are adding a phosphate group to ADP to form ATP, but the energy stored in that terminal phosphodiester bond comes from the photoexcitation of that pair of electrons, and then that energy is transferred to the ADP and inorganic phosphate to form the ATP as that pair of electrons is transferred down the electron transport chain. So this is called photophosphorylation because it is the phosphorylation of ADP to form ATP using energy from sunlight, hence the prefix photo here. So the electron hole at photosystem 2, again, if you have photoexcitation of the 2 electrons in this reaction center chlorophyll molecule, well then you're leaving a pair of electron holes here. Now those are filled because water molecules will donate electrons to fill that hole. So what's happening here is water is being split to form one atom of water, or half a molecule of water, hence the half in front of this O2 here. So H2O is split to form 1 oxygen and 2 protons, but those 2 protons also are accompanied by 2 electrons, so that this equation is balanced both in terms of the atoms on the left and the right hand side, but also the electrostatic charge on the right and the left hand side. So the splitting of water here gives 1 oxygen atom, 2 protons and 2 electrons. Those 2 electrons are then used to fill that pair of electron holes in photosystem 2's reaction center. So the donation of electrons by water, this comes as a result of the splitting of water to form hydrogen ions and oxygen gas, and this is referred to as photolysis of water. So we can see here first of all where the water is used in photosynthesis and where the oxygen is produced. Oxygen gas is actually a waste product of photosynthesis. It is formed as a result of the splitting of water. So the splitting of water to form hydrogen ions and oxygen gas is called photolysis. So now the electron holes at both photosystems have been filled and the process can start all over again. So as the light energy, in other words, the 2 photons, the 2 packets of energy are absorbed by photosystem 2, a pair of electrons are excited to a higher energy level. These electrons then transfer down an electron transport chain, as we saw before, causing protons to diffuse across the thylakoid membranes into the interthylakoid space. Now these protons are then transported down their concentration gradient through an enzyme called ATP synthase. Now this is embedded in the thylakoid membranes, and this creates ATP by direct phosphorylation from ADP to ATP. So the movement of ions, in this case protons, down a selectively permeable membrane or through a selectively permeable membrane down their concentration gradient. Now in this case it's an electrochemical gradient because you have protons massing up on one side. You have a high concentration of protons on one side of the membrane versus the other. So you have a chemical gradient, you have a chemical concentration gradient, higher proton concentration on one side, lower on the other side, but you also have a higher charge, a higher positive charge on one side than the other because you have more positively charged protons on one side versus the other. So it isn't just a concentration gradient, it is an electrochemical gradient. So what we're saying here is that the electrons being transported down the electron transport chain doesn't just directly form ATP. What it does is it causes the diffusion of protons across the thylakoid membrane into the interthylakoid space. Now this sets up a electrochemical gradient of protons across that membrane. So this is essentially what powers the production of ATP. So it relates to the generation of ATP by the movement of hydrogen ions across the membrane during either photosynthesis or respiration. So the name chemiosmosis or chemiosmosis, it derives from the fact that the hydrogen ions diffuse in a way which is similar to how water molecules move across a membrane during osmosis. So again, osmosis is defined as the random movement of water molecules from a region where the water molecules themselves are in higher concentration to a region where the water molecules are in lower concentration through a selectively permeable membrane. Now, this is very similar to what's happening here. The only difference is we're dealing with hydrogen ions instead of water molecules. So photophosphorylation is the production of ATP using the energy of sunlight. Now photophosphorylation is made possible as a result of chemiosmosis as being described here. So essentially photophosphorylation, this process by which ATP is being formed from ADP and inorganic phosphate, this is powered by essentially the electrochemical gradient of hydrogen ions across that membrane, because the transport of electrons down the electron transport chain in the thylakoid membrane leads to the diffusion of hydrogen ions such that you have an electrochemical gradient across that membrane in the grana. Now, this then powers the production of ATP, and so photophosphorylation, this process here, is made possible as a result of chemiosmosis, the diffusion of hydrogen ions across that membrane. So the electrons released from photosystem 1 to the primary acceptors are ultimately passed via ferredoxin to a molecule of the hydrogen carrier NADP. NADP stands for nicotinamide adenine dinucleotide phosphate. Now, NADP is a hydrogen carrier, and again, this is going to be a substance which you're going to become familiar with because we'll find it's mentioned not just in the context of photosynthesis but also in the context of respiration as well. So ultimately, the electrons that find their way to the primary acceptor from photosystem 1 are passed to ferredoxin. Ferredoxin itself then passes those electrons onto a hydrogen carrier, NADP+. So what then happens is that these electrons, this pair of electrons, together with the hydrogen ions which were formed from the photolysis of water, then combine to reduce NADP to NADPH. Now again, here we are using the word reduction because reduction is defined as the gain of electrons by a substance or by a species, a particle, or it can also be used to describe the gain of hydrogen atoms by a molecule or by a particle. Now both of these things are happening in the case of NADP. It's gaining 2 electrons, it's also gaining 2 protons. Now the ultimate outcome of this is that you form one molecule of NADPH, or reduced NADP as it's sometimes called, and one hydrogen ion, one free hydrogen ion. And so the light-dependent phase of photosynthesis has generated ATP and also has generated a molecule of NADPH. Now this NADPH molecule itself has what we call reducing power because it has a hydrogen atom attached to it which can be used to reduce other substances. Now as we'll see, that's going to become important later on. So ATP and NADPH, or reduced NADP, these are both needed to drive the next phase that we're going to be talking about, the light-independent phase of photosynthesis. So let's quickly summarize what we've been talking about. Now this is sometimes called the Z scheme. So we start with the absorption of a pair of photons by both of these photosystems. Now again, all of the molecules shown in this diagram are embedded in the thylakoid membranes of the grana in the chloroplast. So a pair of photons is received by the antenna complexes of both photosystems 1 and 2. This then leads to photoexcitation of the reaction center chlorophyll molecules, each of which release 2 electrons. Those 2 electrons are promoted to a higher energy level and are absorbed by primary acceptor electron acceptor molecules in the thylakoid membrane. Now this accompanies photolysis and the electrons from the photosystem 2 leave a pair of electron holes which are filled by the 2 electrons which are released when one water molecule is split to form an atom of oxygen and 2 protons. In turn, the 2 electrons from photosystem 2 which were accepted by the primary acceptor at a higher energy level are then passed down a chain of successively lower energy level electron carrier molecules in this electron transport chain. This then leads to the diffusion of protons into the interthylakoid membrane space across the thylakoid membrane, forming a electrochemical gradient of protons. That electrochemical gradient of protons is used to drive the photophosphorylation of a molecule of ADP by an inorganic phosphate group to form a molecule of ATP. Those electrons are then passed on to fill the electron hole left by the photoexcitation of the chlorophyll molecule in reaction center of photosystem 1. But the 2 electrons that were excited from this reaction center are accepted by, again, a primary acceptor molecule in the thylakoid membrane at a higher energy level. That pair of electrons is then passed via ferredoxin to an NADP molecule together with 2 protons from the photolysis of water to form a molecule of reduced NADP or NADPH and a free hydrogen ion. So the purpose of the light-dependent stage is to generate both ATP by photophosphorylation and also reducing power in the form of NADPH, which is going to be used, as we'll see in a moment, in the light-independent reactions. So we've looked at the light-dependent reactions. Let's take a look now at the light-independent reactions. Okay, so before we go into the light-independent reactions now, I need to quickly highlight one thing, which is somewhat of an extension of the GCSE understanding of photosynthesis. So at GCSE level, you will have come across the overall balanced symbol equation for photosynthesis. Now, I'm sure many of you will have already worked out at this point that that's a massive oversimplification. In fact, photosynthesis and also respiration, as we'll see later on, even though they are described in terms of a single overall balanced symbol equation, that symbol equation is somewhat misleading and oversimplistic because it implies that the processes that we're talking about, photosynthesis and respiration, happen in one chemical step. That is not the case. So the photosynthesis overall equation is usually summarized like this, like you've seen before, and this overall shows that there are 6 molecules of carbon dioxide, 6 molecules of water required to produce 1 molecule of glucose and 6 molecules of oxygen. Now, this familiar summary equation implies, first of all, that it all happens in one chemical step, which is, of course, not the case, but it also hides a sequence of reactions in between, going from the reactants to the products. Many separate reactions happen with many intermediates, but also a number of other molecules which are involved. And these reactions are driven by ATP and also NADPH, reduced NADP, as we saw before. So the precise biochemical reactions in which these 6 molecules of carbon dioxide are used, this is known as the Calvin cycle. So Melvin Calvin and his co-workers determined the chemical steps involved in the reduction of carbon dioxide to carbohydrates during the light-independent stage of photosynthesis, and for this he and his co-workers were awarded the Nobel Prize in 1961. Now, the experimental procedure used by Calvin et al. Involved the use of radioactive form of carbon, which is carbon-14. Now, we're going to go into the biochemical, or rather the experimental evidence underlying our understanding of the biochemistry of photosynthesis in the next lesson of this sequence, but for now it's enough to say that the way that they did this was by providing cultures of single-celled photosynthetic algae with radioactive carbon or radioactive carbon dioxide containing carbon-14, and then using radiography to follow what happens to that radioactively labeled carbon as it was assembled into glucose by the algae through the process of photosynthesis. So let's have a look at the light-independent stage of photosynthesis, particularly the Calvin cycle. Now, we're going to need to break this down. Again, these are arbitrary stages that we're breaking these processes down into for the purposes of discussing them and talking about them. But essentially, we can talk about the first stage of the Calvin cycle as being the fixation of carbon dioxide. So the light-independent stage of photosynthesis takes place, first of all, not in the thylakoid membranes as we saw for the light-dependent stage. Instead, this takes place in the stroma of the chloroplast. Now, the first stage of the Calvin cycle we can describe as carbon dioxide fixation. Now, located within the stroma of the chloroplast is a 5-carbon phosphorylated sugar known as ribulose bisphosphate. Now, ribulose bisphosphate, again, you don't need to necessarily know the structure of ribulose bisphosphate molecules. It's enough to track the number of carbons and the number of phosphate groups involved in these molecules as we go around. So don't worry too much about the structures of ribulose bisphosphate or any of the structures in the Calvin cycle. It's enough to just know how many carbons are involved in them, what their names are, and their relationships to other substances. So this simplified diagram illustrates that ribulose bisphosphate is a 5-carbon sugar with 2 phosphate groups. So this is found in the stroma of the chloroplast. Now the enzyme ribulose 1,5-bisphosphate carboxylase, sometimes called oxygenase, is usually abbreviated to Rubisco. Now this enzyme catalyzes the reaction between a molecule of carbon dioxide and this 5-carbon phosphorylated sugar, ribulose bisphosphate. This is called carbon dioxide fixation because it's this step of the light-independent reactions which involves taking the carbon dioxide absorbed by the plant in the leaves and fixing it, actually putting it into a molecule which is going to be used in this process of photosynthesis. So this reaction ultimately forms an unstable 6-carbon compound, and it's unstable and therefore breaks down immediately to form 2 molecules of a 3-carbon phosphorylated compound called glycerate 3-phosphate. Now glycerate 3-phosphate, again, you don't need to know the molecular structure of it. It's enough to know that this molecule here combines chemically with 1 molecule of carbon dioxide, or the carbon from 1 molecule of carbon dioxide, forming an unstable 6-carbon intermediate, which then breaks down immediately to 2 molecules of glycerate 3-phosphate, shown here. So glycerate 3-phosphate is the first stable compound to be formed during the Calvin cycle. So we have here carbon dioxide fixation, the first stage of the Calvin cycle. Now the next stage of the Calvin cycle involves the reduction of each one of those molecules of glycerate 3-phosphate to the phosphorylated 3-carbon sugar triose phosphate. And so this is shown here. Now I'm showing the molecular formulae of these compounds just to give you a sense of the numbers of atoms of each element involved. So this is slightly more detail that is being provided by these diagrammatic forms here. These forms simply give you a sense of the number of carbon atoms and the number of phosphate groups. These are more accurate in the sense that they give you a sense of the molecular formulae. Now again, you don't need to know the molecular formulae. This is just to aid your understanding of what's going on. So this reduction step, and again, look at the terms being used here. Reduction. Reduction can be defined in 3 different ways in chemistry and in biology. Reduction is the gain of electrons, it's also the gain of hydrogen atoms, and it's also the loss of oxygen atoms. And correspondingly, oxidation is the loss of electrons, or the loss of hydrogens, or the gain of an oxygen atom. So if we look at what's happening here to the molecular formulae of each one of these glycerate 3-phosphate molecules, we see that we are going from C3H7O7P to C3H7O6P. So in this sense, these molecules are both being reduced because they are both having an oxygen atom removed from them. So this is the reduction of glycerate 3-phosphate to triose phosphate, and again, because we started with 2 molecules of GP, we end up with 2 molecules of TP. So this reduction step requires the hydrogen from NADPH. So again, this is where that NADPH, that reduced hydrogen carrier, or the reduced NADP from the light-dependent stage, comes in. And this is also an endergonic reaction, a reaction that requires an input of energy from the environment. And so this requires not just an NADPH molecule to provide the hydrogen necessary to remove this oxygen from each one of these, it also requires the hydrolysis of ATP. Now because we're doing this for 2 molecules, it requires 2 molecules of ATP to be hydrolyzed to release the energy required for this step. It also requires 2 molecules of reduced NADP or NADPH to be oxidized, to have their hydrogens removed from them. So this is the second step, the second stage of Calvin cycle. So the conversion of GP to TP requires the products of the light-dependent stage. We need 2 molecules of NADPH, and we also need 2 molecules of ATP. Both of these were produced in the light-dependent stage. So the NADP plus produced in the formation of TP here can then be reduced again in the light-dependent stage. So these processes are related to each other, as we'll see later on. So we get the NADPH and the ATP from the light-dependent stage to do this step, this second stage of Calvin's cycle, but in turn the NADP here and also the ADP and inorganic phosphate can then be cycled back round to that Z scheme that we saw before for the light-dependent stage in order to reform the ATP and the NADPH needed for another cycle of Calvin's cycle. So the third stage of Calvin's cycle is the regeneration of ribulose. This is a cycle, so we need to somehow get from here back to our starting point. So as the triose phosphate molecules accumulate, some of these are used to form glucose, which can then be used either in respiration or turned into polysaccharides such as starch and cellulose and so on. But also some of it is cycled back round to reform ribulose bisphosphate. Now again, it's not just starch and cellulose which we can use glucose for, or which plants can use glucose for. Plants need glucose to synthesize the other monomers of their biological polymers, things like amino acids and also fatty acids. So some of the triose phosphate molecules are used for these purposes here, but some of them are then used to regenerate the compound ribulose bisphosphate to enable this cycle to complete and continue to operate. So the way that this is done, as we'll see in a moment, is that this stage utilizes again ATP for the phosphorylation of ribulose phosphate, which is formed from some of these triose phosphate molecules, to ribulose bisphosphate. So ribulose phosphate is formed from some of the triose phosphate molecules, but in turn this needs to be phosphorylated one more time to make ribulose bisphosphate. And the phosphate group and also the energy for this process comes from a molecule of ATP. So this is Calvin's cycle. Again, this is a simplification. It is more complex than this, and we are simplifying significantly the structures of these molecules, but this should be more than enough for most A-level biology curricula. Now, if you want more detail on this, please do send me a message via the student notes tab on this lessons page, or if you'd like to engage in a discussion with other students and have me weigh in as well, you can write your questions on in the comments tab or in the live chat tab for this lesson, and I'll be happy to answer your questions there. So let's summarize Calvin's cycle. So we start with ribulose bisphosphate, one molecule of this, combining with one molecule of carbon dioxide in the carbon dioxide fixation stage. This forms an unstable 6-carbon intermediate, which immediately breaks down to 2 molecules of glycerate 3-phosphate. We then have the conversion of these 2 molecules of GP to 2 molecules of triose phosphate, 2 3-carbon phosphorylated sugars, by means of the use of 2 molecules of ATP and their hydrolysis to form 2 molecules of ADP and 2 inorganic phosphate. But also the energy released by this is used to drive this process, because this step, this second reduction stage, is endergonic. It requires an input of energy. In turn, also, this requires the reducing power provided by 2 molecules of reduced NADPH. These then are oxidized. They give up their hydrogens to strip the 2 oxygen atoms from these 2 glycerate phosphate to form 2 triose phosphate molecules. Now, some of these triose phosphate molecules are used to synthesize various necessary substances, such as polysaccharides like starch for storage, cellulose for structure, and also amino acids, fatty acids, and other necessary substances in the plant. But some of them are used to form ribulose phosphate, which is a 5-carbon phosphorylated sugar, but then this is then used to reform ribulose bisphosphate by the hydrolysis of another molecule of ATP to provide both the energy and also the additional phosphate group that you need here. So ultimately, 6 molecules of carbon dioxide were present in the overall summary equation, and in the previous scheme that you saw in that previous slide, only one molecule of carbon dioxide was used. So 6 molecules of CO2 will combine with 6 molecules of RuBP, ribulose bisphosphate. Now again, ribulose bisphosphate is a 5-carbon sugar. We're not showing the phosphate groups here, we're just counting the numbers of molecules of ribulose bisphosphate. So, ribulose bisphosphate, each one of these molecules is a 5-carbon molecule. 6 molecules of CO2 are required to turn these into, again, 6 molecules of a 6-carbon intermediate which is unstable and breaks down immediately. Now, each one of those 6-carbon intermediates will break down to 2 molecules of glycerate 3-phosphate. So here you have a total of 12 molecules of glycerate phosphate being produced by the combination of 6 molecules of ribulose bisphosphate and 6 molecules of CO2. So here's where the 6 molecules of CO2 go in. These are the 6 molecules of CO2 that appear in the overall summary equation on the left-hand side. So the 12 molecules of glycerate phosphate are then reduced to 12 molecules of triose phosphate. Now again, if we multiply up from the previous diagram, this will require 12 molecules of ATP to provide the energy, and also 12 molecules of reduced NADP or NADPH to provide the hydrogen atoms necessary. This then forms 12 molecules of the 3-carbon triose phosphate. So now from this stage, the 12 ATP and again the 12 NADP are required for this step. Now, of this 12 molecules of triose phosphate, 2 molecules of triose phosphate are used to form 1 molecule of glucose. Now again, if you think about this, it kind of makes sense because glucose is a 6-carbon sugar. Each one of these triose phosphate molecules is a 3-carbon molecule, and so you'll require 2 of those to form 1 glucose molecule. Now the remaining 10 molecules of triose phosphate are then used to regenerate 6 molecules ultimately of ribulose bisphosphate. So let's see how that works here. So you take the 10 remaining molecules, you then form 6 molecules of ribulose phosphate. So 12 molecules of triose phosphate, you take 2 of those and you make 1 molecule of glucose. 10 of those, containing a total of 30 carbon atoms, are then changed chemically to form 6 molecules of the 5-carbon sugar ribulose phosphate. So we see here that 30 carbon atoms in the form of 10 of these triose phosphate molecules form 6 molecules of this 5-carbon ribulose phosphate. To reform the 6 molecules of ribulose bisphosphate now, we need to phosphorylate each one of these one more time, and this of course will require 6 molecules of ATP. And so each ribulose phosphate, RUP molecule, is phosphorylated by a single molecule of ATP. So for 6 of these, you need 6 of these, and you're going to form 6 ribulose bisphosphate molecules. And again, at this point, the cycle can form again. So this is a summary which shows the actual numbers of molecules of each one of these stages, at each one of these stages, which correspond to the 6 molecules of carbon dioxide which go into each stage or each cycle of this. Now again, this describes the formation of 1 glucose molecule. So this explains why you have 6 molecules of CO2 which go into photosynthesis versus 1 molecule of glucose. So this is basically the scheme that you need to know. So again, we've divided Calvin cycle into the carbon fixation stage, the reduction stage, which turns our molecules of GP, glycerate 3-phosphate, into molecules of triose phosphate. Again, for every 1 molecule of glucose, you start with 6 molecules of carbon dioxide in the fixation stage. You then end up with 12 molecules of glycerate 3-phosphate, which are transformed into 12 molecules of triose phosphate by the use of 12 molecules of ATP, 12 molecules of NADPH. Both of these come from the light-dependent stage. This then forms 12 molecules of triose phosphate, of which 2 of those form 1 molecule of glucose. The remaining 10 are used to form 6 molecules of ribulose phosphate, which are then phosphorylated by the use of 6 ATP to form 6 molecules of ribulose bisphosphate in the ribulose regeneration stage. So only 2 out of 12, or 1/6 of the triose phosphate molecules, are actually used to form the 6-carbon product of photosynthesis. In other words, only 10 of the 12 are used to form glucose. The remaining, rather two of the 12, the remaining 10 are used to reform ribulose phosphate. So, throughout the course of his experiments, Calvin measured the relative amounts of glycerate phosphate and Ruby P present first in continuous light conditions and then when light was switched off. So, if we look at the relative amounts of substances on this axis and then examine them under conditions of light and darkness, this is what Calvin found. So, in the light, both glycerate phosphate and Ruby P remain constant. But then, as soon as the light is turned off, again, as we saw in the previous scheme, ribulose bisphosphate is converted into glycerate 3-phosphate, which in turn is converted back to Ruby P during the ribulose regeneration phase. So in darkness, the GP accumulates and the levels of Ruby P will fall. So this provides evidence for the fact that the Calvin cycle is disrupted in darkness. And so the effect is observed because the reduction of GP to TP requires ATP and NADPH from the light-dependent phase. So the formation of triose phosphate is prevented, and the regeneration phase of Calvin cycle, where Ruby P is reformed, can't take place under those conditions. So glycerate 3-phosphate will accumulate and ribulose bisphosphate will fall. So Calvin's cycle is dependent. Even though it is the light-independent stage, Calvin's cycle itself is dependent upon the light-dependent stage for its operation. So, to summarize both the light-dependent and the light-independent phase and the way in which they are dependent upon each other, we can take a look at this diagram. So this is a kind of simplified diagram of a chloroplast. So this box represents a chloroplast. On the left-hand side of this box, this represents the processes happening in the grana and in the thylakoid membranes of those grana. And then on the other side of this dotted line here, this represents the processes happening in the stroma of the chloroplast. So we have photosynthesis here as a two-stage process. During the light-dependent stage, it generates ATP and NADPH and then utilizes those products to reduce carbon dioxide to carbohydrate in the light-independent phase. So in the light-dependent phase, you've got light energy coming in, it's being used to synthesize ATP and NADPH. Those then go into the stroma where they can be used to reduce carbon dioxide to carbohydrate. So, on the other side though, when these have been used, again, they are cycled back for use in the light-dependent phase. So we see here the NADP and the ADP and inorganic phosphate are cycled back to the light-dependent phase so that they can be used to form more NADPH and ATP. So water is used, remember photolysis happens in the light-dependent phase and oxygen is produced as a consequence of photolysis, whereas carbon dioxide goes into Calvin's cycle in the light-independent phase and it's the sugars which are formed, glucose being formed from two molecules of triose phosphate in the light-independent stage. So again, this diagram is kind of illustrative of the processes, where they occur, and the connectivity between those processes as well. So, we've talked about the biochemistry of photosynthesis, the light-dependent and the light-independent phase. What we now need to do is to take a look at the factors which will influence the rate of photosynthesis and the way in which those factors can become limiting to that rate. So this is essentially going to be an extension of what you should have already come across at GCSE level. So the three most important factors which influence photosynthesis and the rate of photosynthesis are the three being showed here: the intensity of the light, the temperature, and the concentration of carbon dioxide. So the rate of photosynthesis is affected by these abiotic environmental factors, as we've seen. Now, the rate at which it happens is limited by whichever one of these factors is in the shortest supply, and this is essentially the law of limiting factors. So, the law of limiting factors can be stated by saying that when a process is affected by a number of variables or factors, then the factor which is in shortest supply at a given time is going to be the one that determines the overall rate of the process. Now, this is a law which doesn't just affect or doesn't just dictate the way in which photosynthesis is affected by these factors. It's also a law which applies to many other biochemical processes which are dependent on a number of factors. So at certain times of day, for example, light intensity is the limiting factor for photosynthesis. So in other words, light intensity, or the lack of available light, will determine the rate of photosynthesis, given that the other necessary variables, things like temperature and carbon dioxide concentration, are not limiting. So if the limiting factor is increased, like if the light availability is increased, then the rate of photosynthesis will correspondingly increase until one of the other factors limits it. So the following graph shows the effects of changing the light intensity on the rate of photosynthesis. So if we measure the rate of photosynthesis on this axis here, and the increasing light intensity on this axis, we see that at low levels, increasing the rate of or increasing the light intensity will increase the rate of photosynthesis rather rapidly. So in this phase, light is the limiting factor in this region of the graph, because increasing the light intensity is having a corresponding effect, an increase in the rate of photosynthesis. So an increase in light intensity can result in an increase in the rate of photosynthesis only when light is the limiting factor. Now, when you get to this point, increasing the light intensity further from here onwards has no effect on the rate of photosynthesis. So at this point, some other factor has become the limiting factor. So increasing the light intensity will increase the rate of photosynthesis, but at higher light intensities like these, increasing the light intensity has no effect on the rate of photosynthesis, because at this point some other factor, such as carbon dioxide concentration or temperature, is limiting the rate of photosynthesis. Now, the effect of varying the temperature in the carbon dioxide concentrations can be illustrated on this graph here. So we're going to look at a couple of different experiments here which illustrate the independent effects of changing both the carbon dioxide concentration and the temperature on the rate of photosynthesis at differing light intensities. So the first experiment illustrates that temperature and carbon dioxide concentration are both limiting at higher light intensities here. So initially at low light intensities we raise the light concentration, and this will increase the rate of photosynthesis up to this point here, up to this light intensity here. At this point, it's some other factor affecting it. Now, this experiment was done at 15 degrees centigrade and at 0.04 kilopascals of carbon dioxide. This is the partial pressure of carbon dioxide at which this first experiment is done. Now, if we look at the effect of changing just the temperature and keeping the carbon dioxide concentration the same, and then again running the experiment again and measuring how the rate of photosynthesis changes with light intensity, we would get the following curve. So we see here that increasing the temperature from 15 degrees C and 25 degrees C and keeping the carbon dioxide concentration the same results in an increased rate of photosynthesis, which continues to increase beyond the point where the other factors affected it in the previous experiment. So now if we then increase the CO2 concentration from 0.04 kilopascals to a higher concentration, we can see the effect of that in experiment number 3. Here we've kept the temperature the same, but we've changed the carbon dioxide concentration. So we can see here that both carbon dioxide concentration and temperature are limiting the rate of photosynthesis in experiment 1, but increasing either of these factors, whether we choose to increase just the temperature or we choose to increase the carbon dioxide concentration, both of these will increase the rate of photosynthesis, with the greatest increase occurring when both of these values are increased, as in experiment 3. So we can conclude here that at high light intensities, both temperature and carbon dioxide concentration become the limiting factors. Therefore, an increase in either factor results in an increased photosynthetic rate, and the greatest increase is seen when both of those factors are increased. Now, commercial growers will make use of the law of limiting factors and their understanding of how that applies to photosynthesis in order to maintain optimum conditions in their greenhouses to produce the highest yield of flowers or of fruits or of vegetables that they can. So, Artificial lighting can be used to increase the light intensity, although again care has to be taken to ensure that the light intensities are not too high where they would bleach or damage the chlorophyll in the plant leaves. You can increase the availability of carbon dioxide either by using heaters that burn fossil fuels or by pumping carbon dioxide gas into the greenhouses. Again, the heating will in turn increase the temperature, and this again will prevent the night temperatures in those greenhouses from falling too low, and also optimizes the temperature at which those plants will photosynthesize most effectively. So, we now need to expand our understanding a little bit beyond GCSE, because what we've done there is talk about the ways in which those limiting factors affect carbon dioxide concentration. Now, essentially everything that we've said there in the previous section of this video has essentially been at GCSE level. The only real thing that goes a little bit beyond that is our use of specific values and looking at the ways in which changing those values changes the rate of photosynthesis. But at advanced level, we need to talk a little bit in a bit more detail about compensation points, which is perhaps something that you haven't come across at GCSE level. So in photosynthesizing organisms, it's important to remember that these are organisms, and all living things, do respiration. Respiration is a cellular process, which is one of the 7 characteristics of all living things. So in photoautotrophs like plants and green algae, it's not just photosynthesis that's taking place, it's also respiration. Now, both of these processes involve gas exchange with the environment. Now, during daylight hours, what this means is that you've got the light-dependent reactions going on, and therefore green plants are both photosynthesizing and respiring. Now, during the night period, green plants only respire. So as the light intensity begins to increase from dawn, there comes a point where the rate of respiration is equal to the rate of photosynthesis. So the light intensity at which this happens, the light intensity at which the respiratory rate is equal to the photosynthetic rate, this is called the light compensation point. And the time taken for a green plant to reach this point after having been in complete darkness is called the compensation period. So at the compensation point, carbon dioxide output is equal to carbon dioxide uptake, such that there is no net loss or gain of that gas. So this is how we define the terms compensation point and compensation period. Now, before we go on, a lot of students get confused about this because they say, well, surely during night time there's the light independent reactions going on. They've kind of confused the term light independent with darkness. Light independent simply means that phase of respiration, that phase of photosynthesis rather, those biochemical reactions do not rely upon light input. That doesn't necessarily mean that they occur in darkness. It's slightly more nuanced than that. So again, think of light dependent versus light independent, not in terms of happening in light and happening in darkness, but in terms of the light dependent stages are the ones that use light energy. The light independent stages are the ones that don't need light energy, and you won't get too confused there. Again, if you have any questions on that, please do either write them as comments in the live chat or send me a private note via either your private coaching area or through the student notes function on this lessons page on my website. So we talked a little bit in the previous lesson about the adaptations of sun and shade plants on a basic level. We now need to look at the specific adaptations that these organisms have. So sun plants thrive in conditions, again, where light intensities are relatively high, whereas shade plants typically are found in more dimly lit habitats. Now, typical woodlands will show levels of light intensity or stratification, and basically this arises because the canopy of the trees, the tallest organisms in that environment, will typically dominate the upper woodland areas, and this then kind of allows less light energy to filter down to the lower levels of that woodland. So many of the woodland perennials will occupy the lower shaded herb layers of the woodland. So the sun plants are things like these very tall trees. These have unrestricted access to the level of light intensity coming from the sun, and therefore these are organisms which are sun plants. They're described as sun plants because they are their leaves are exposed to high light intensity layers of the woodland. Now, in contrast, this shade plant, oxalis, which is found in the lower herb layer of the woodlands, this is a shade plant. This is adapted to dimmer light conditions because it's overshadowed by these taller plants. So investigations into the carbon dioxide uptake and output for sun and shade plants have been performed, and we can see some of the results from these here. So on the horizontal axis here, we're measuring light intensity in a unit called lux. Now, on the vertical axis, we have positive values corresponding to net CO2 uptake by plants versus negative values corresponding to net CO2 release. So the negative values correspond to when respiration is happening at a higher rate than photosynthesis, and the net CO2 uptake, the positive values, correspond to the opposite, when the rate of photosynthesis is higher than the rate of respiration. So whenever you have 0 on the vertical axis, the rate of photosynthesis is equal to the rate of respiration, and so there is no net uptake or release of CO2 by the plant. So if we examine this curve for sun plants, we see a curve like this, whereas for shade plants, we see a curve like this. So we can see here that the compensation point for the shade plant occurs at a lower light intensity to that of the sun plant. Now, the shade plant, the reason for this is because the shade plant begins to photosynthesize at a much lower light intensity than the sun plant. So the compensation point for the shade plant is also much shorter than that for the sun plant, or the compensation period rather. Now what this means is the shade plant takes a shorter time to reach its compensation point than the sun plant does. So these would correspond to the compensation periods. So the ability of the shade plants to make use of the low light intensities for photosynthesis obviously has survival value for these species. Now, they have various other adaptations to tolerate the dimly lit habitats. We mentioned these before. These include their leaf structure and the fact that also they flower and seed earlier in the year before that dense tree canopy can form, which overshadows them. So these are some wild garlic plants. These are adapted to thrive in shady conditions, and the bulb plants flower and seed in the spring before the tree canopy comes into full leaf and overshadows them. So before we finish, you've probably gotten used to me providing you with some extension work to extend your understanding, and this lesson's going to be no different. So let's have a look at some further ideas here that will extend your understanding a little bit further. So we talked about wavelengths of light. Now, absorption spectra are actually quite important in the context of photosynthesis and respiration. So when a beam of light is passed through a glass prism, the visible spectrum of white light is obtained by the different levels of refraction of the different wavelengths. In other words, the different wavelengths that compose white light are then split into the separate colors or the separate frequencies or wavelengths that make them up. So this can happen when you get a beam of light which then passes through a solution of leaf extract. Now, when we do this, we can obtain what's called an absorption spectrum. So basically, we're making use of the principle that white light is made up of the full range of visible wavelengths. We're passing that through a solution of leaf extract containing all of the photosynthetic pigments found in a leaf, and then we are splitting that light coming through that leaf extract using a prism to find out which wavelengths pass through or are transmitted and which wavelengths have been absorbed by those pigments. And this is what you might obtain, what's called an absorption spectrum. So the black bands will appear in the spectrum where particular wavelengths of light have been absorbed. Now, predominantly, the wavelengths at the blue and the red end are the ones that have been absorbed, as you can see here. So what this tells you is that it's predominantly the blue and the red wavelengths which plants make use of for photosynthesis. So the spectrum obtained by passing white light through a leaf pigment extract shows us the wavelengths that the pigments have absorbed, and more detailed information about the absorption processes can be obtained by using a device called a spectrometer. So the degree of absorption, the extent to which those wavelengths have been absorbed, can be measured for both the total plant extract and also for the individual pigments. So what we did in the previous example was just simply take the extract from the leaves of a plant containing all of those photosynthetic pigments and have a look at the wavelengths that have been absorbed by all of those pigments together. But this doesn't tell us much about what wavelengths those individual pigments will absorb. To do that, we need to separate those pigments by, for example, chromatography, and then do the same experiment again. So this information can then be used to generate what's called an absorption spectrum graph. Now, in a similar way, the effectiveness of different wavelengths of light in bringing about photosynthesis can also be determined. So the rate of photosynthesis can be measured at different wavelengths of illumination, or using different wavelengths of light to illuminate the plants. So that information can also be used to generate a different kind of graph called an action spectrum graph. So we can obtain an absorption spectrum graph by having a look at the wavelengths of light which are absorbed by the photosynthetic pigments in a particular graph. But we can also find out which wavelengths of light result in the most rapid rate of photosynthesis, and we can obtain an action spectrum graph from that. So we can then go on to compare the action spectra and the absorption spectra to find out exactly how these different wavelengths and the pigments which absorb them play a role in photosynthesis. So here we have wavelength on this axis in nanometers, and we have two axes on this graph here because we're going to show two curves. We're going to show the percentage absorption by the pigments of the plant of these different wavelengths, and we're also going to show the rate of photosynthesis of the plants when illuminated by monochromatic light of these different wavelengths. So this here is the absorption spectrum. This shows us that these pigments will absorb most strongly in the blue end of the spectrum, and the second strongest absorbance is in the red wavelengths. Now we can see here the green wavelengths in the middle are not absorbed. In fact, these are the ones which are predominantly reflected by the plant, and this is why many plants appear green. Now if we compare this to the action spectrum, we can see this curve here, and we can see that the action spectrum actually matches quite closely the peaks of the absorption spectrum. So although all wavelengths between 400 and 700 nanometers are absorbed to some extent by the plant, the maximum absorption occurs in the blue and the violet, and also in the red regions of the spectrum. So there's a close correlation here between the action spectrum and the absorption spectrum. Now the rate of photosynthesis is largely determined by the amount of light of different wavelengths being absorbed. Green light is not absorbed, it is reflected predominantly, and this is why most leaves are visibly green. Okay everyone, that's going to do it for this lesson. The next lesson in this sequence, we are going to go into details about the experimental evidence which supports our understanding of the biochemistry of photosynthesis that we've talked about in this lesson.